Hi, everyone. As part of this presentation, we'll be covering the very interesting, yet often quite challenging topic of cleaning validation. As part of this presentation, we'll be going through a number of topics relevant to the cleaning validation, such as the cleaning validation lifecycle approach, the application of quality risk management, an overview of different types of residues, approaches towards setting acceptable cleaning limits for equipment surfaces. We'll also taking a look at common cleaning methods, approaches towards sampling and testing, as well as the application of the matrix approach in multi-product facilities. Finally, at the end of this presentation, we'll be taking a look at the number of deficiencies that have come up in recent HPRA inspections. By definition, cleaning validation is the systematic approach of generating documented evidence that an approved cleaning procedure can effectively remove the residues from the product contact surfaces, such as APIs, cell debris, cleaning agents, and other contaminants down to an acceptable predetermined level, so as to prevent potential cross-contamination and thus ensuring the quality of the next product remains integral. Cleaning validation is often approached as a discrete kind of exercise. However, at this point, it has to be stressed that cleaning validation is not monolithic and should be approached through the lens of a life cycle approach. In line with the life cycle approach as per Annex 15 on qualification and validation, cleaning validation encompasses the initial process design, followed by the performance qualification or the validation of the process, supplemented by an ongoing periodic revalidation, also known as continued verification stage. This is, of course, yet another oversimplification of the cleaning validation lifecycle, which in reality tends to be far more complex, involving a number of steps such as cleaning development, cycle development of the cleaning method, recovery studies for rinse and swap sampling, analytical method validation, various risk assessments, the actual cleaning validation, followed by periodic revalidation of the remainder of the product's life cycle. As it stands, a risk-based approach is necessary to assess and contain risk associated with cleaning validation, especially in multi-product facilities where the risk of cross-contamination potential is far greater. A preliminary risk assessment should therefore be performed to determine all contributing factors which may influence the effectiveness of cleaning procedures and provide an outline for the cleaning validation approach. As the extent of the quality risk management process is proportional to the underlying risk, greater emphasis should be placed in areas such as determining the nature of the residue and the solvent or detergent, if necessary, needed to remove the residue, taking into account the equipment design, material of construction, as well as identifying the difficult to clean locations. Grouping of products and equipment and validating for worst-case combinations may be considered. Also, the level of automation that the company has at their disposal, as opposed to relying completely on manual cleaning processes, should be considered, as well as the possibility of cleaning in place or requiring a certain level of equipment disassembly to clean out of place must also be taken into account. Time is another key consideration that needs to be accounted for as part of the QRM assessment in preparation for cleaning validation. The maximum length of a campaign, the dirty hole times and the clean hole times, must be considered, but also the processing times that take to effectively remove residues. The risk assessment that is performed as part of the quality risk management is a living document and should be revisited and reevaluated as a better understanding of the cleaning validation process becomes available. Depending on the level of risk, it may even be necessary to dedicate premises and equipment for manufacturing and/or packaging operations to control the risk presented by some medicinal products, especially where cleaning processes are ineffective or not appropriate to be used on certain pieces of equipment. Every cleaning validation program should be based on the identification and evaluation of potential residues and assigning of appropriate acceptance levels. Typical residues include APIs, which can be either small molecules or macromolecules, excipients, degradation byproducts, cleaning agents, bioburden, and toxin. Residue levels must maintain pharmacological safety and stability while avoiding toxicity or contamination of the product that follows. Before validation studies can begin, cleaning cycle development studies must be performed to define the conditions necessary to clean product residue effectively down to an acceptable level. Validation should also take into account the level of automation in the cleaning process, whether equipment design allows for cleaning in place, or whether cleaning the equipment out of place is required. The specified normal operating range of utilities and equipment should be validated. The number of cleaning cycles that are necessary to remove the residue, the type of equipment, which includes the critical process parameters, as in the detergent type and concentration selection, the temperature of cleaning solution and the cleaning action, detergent contact and rinsing times, as well as the number of cleaning cycles required. The conditions established during cleaning development should be confirmed under an executed protocol prior to validation activities. Once the residue has been identified, effective removal of it should be demonstrated. This is supported by setting appropriate limits to support visual cleanliness, as in no visible residues on the surface should be present, microbiological cleanliness, as in the absence of bioburden endotoxin, chemical cleanliness, as in the effective removal of APIs, excipients, and detergents. The determination of cleaning validation limits must be scientifically justified and based on toxicological evaluation. Residue levels must maintain pharmacological safety and stability while avoiding toxicity or contamination of the product that follows. Historically, a number of methodologies have been employed for the determination of residue limits of product A finding its way into a subsequent product B. And these have included the 10 particles per million criterion, which corresponds to the allowance of 10 milligrams of hazardous contaminants contained into 1 kilo of a product, the LD50 approach, which corresponds to the lethal dose, which, if given all at once, causes the death of 50% of individuals of a group, and then the 1,000th of a dose method that relies on the assumptions that 1,000th of a dose of any product appearing on the next one will impart limited or no pharmacological effect on the subjects. However, relying on these approaches obscures the true patient risk and presents a potential compliance gap, which is why these approaches must be abandoned in favor of the health-based exposure limits approach based on the permissible daily exposure approach. In November of 2014, the EMA issues the guideline on setting health-based exposure limits for use in the risk identification in the manufacture of different medicinal products in shared facilities. The general principle of which was based on the absence of adverse effects on patients, whether pharmacological or toxicological. Unlike the approaches listed in the previous slide, a more scientifically backed approach is proposed for the determination of the health-based exposure limits based on the permissible daily exposure limit, PDE for short, that represents a substance-specific dose that is unlikely to cause an adverse event to individuals if exposed to that dose daily over the course of a lifetime. The PDE-based HBL's calculation takes into account a number of factors, such as the no observed adverse effect limit, several adjustment factors to account for various uncertainties, but also weight adjustment for the individuals that receive the residue. Once the health-based assessment has been completed and the HBL confirmed, they should be used as part of the quality risk management process. This QRM should be carried out prospectively in the case of new equipment or facilities to assess if the existing organizational and technical control measures are adequate and to determine what controls are needed to be applied to minimize the risk of cross-contamination. One might wonder, however, what practical use do PDE values have in the context of clean validation? Well, PDE are used in the maximum allowable carryover limit determination, which corresponds to the fraction of product A that could end up in the subsequent batch of product B. In this example taken from the ISP guide, the determination of the maximum allowable carryover limit takes into account a number of factors, such as the permissible daily exposure limit of product A, which is derived from toxicological data, the minimum batch size of the next product B, which determines the degree to which any carryover is spread among the subsequent product dosages, and then the maximum daily dose of a product B, which might be administered to a patient. However, as sampling and testing the equipment to determine the amount of residue present on it would be practically impossible, Macro levels may be adjusted by taking into account only a small area on the equipment surface to be sampled over the total cumulative area of the equipment train. That would enable us to relate the result of a single sample to the total residue amount in the manufacturing equipment. Also, the impact of recovery factors derived from the recovery studies, which we'll discuss in the following slides, must be taken into account either by incorporating them into the macro calculation or in the analytical method calculations. Although the respective EMA guideline on the determination of the health-based exposure limits may be used to justify cleaning limits, other approaches could be accepted as long as they are adequately justified. For example, in the case of therapeutic macromolecules, setting HPL-based PDE limits corresponding to active and intact product may not be required, as therapeutic macromolecules are prone to degradation and denaturation when exposed to extreme values of pH and heat that renders them pharmacologically inactive. Another example would be that of existing products, where the manufacturer's historically used cleaning limits should be retained and considered as alert limits, provided that cleaning process capability has been taken into account. Once companies have determined the list of residues and other contaminants, a suitable detection method needs to be selected to screen for those residues. Typically, the selection of analytical methods for the detection of the identified contaminants boils down to whether a pharmacological assay exists, a pharmacopoeial assay exists, whether to be opting for a specific or non-specific methodology, and whether the selected method is sufficiently capable to detect the contaminants in terms of its inherent limit of detection or limit of quantitation thresholds. Specific methods for an individual ingredients include high-performance liquid chromatography HPLC titration, whereas non-specific methods include TOC, total organic carbon, pH levels, and conductivity. Once the worst-case product has been determined and the macro levels for the product defined, and an appropriate analytical method for the detection of the residues has been selected, clinical validation studies may then commence to evaluate the effective removal of product residues from the equipment surface. The conditions established during cleaning method development should be confirmed under an executed protocol in the validation studies. Typically, in a cleaning validation campaign, soiled equipment after a production run must be cleaned with the cleaning method undergoing validation, and cleaned equipment must be sampled to determine whether any remaining residue is below the defined macro levels, which, if successfully demonstrated over a number of runs, will render the cleaning method validated. Successfully validated cleaning methods are the subject to further risk assessment to determine the frequency of periodic revalidations in order to support the validated status of a method over the lifetime of the product, process, and equipment. Failure at the cleaning validation stage, however, should warrant further investigation through the deviation process, and if not adequately resolved, the companies might consider going back to the drawing board to further tweak the cleaning method in question. Through post-cleaning sampling, an estimate of the amount of residue of the equipment might be achieved. To facilitate that, a number of sampling methods are applicable, such as rinse sampling or swab sampling. Sampling should be carried out by any of the proposed sampling methods, depending on the nature of the equipment, the residues being measured, the residue limit, and the analytical method at hand. Cleaning validation protocols should specify or reference the locations to be sampled, the rationale for the selection of these locations, and define the acceptance criteria. Swab sampling is used to collect residues directly from the surfaces for further analysis. It is the preferred sampling technique due to the ability to sample hard-to-clean critical locations. Similarly, just like swab sampling, rinse sampling is another sampling method which is preferred for sampling large pieces of equipment or runs of piping and generally locations inaccessible to swabbing. This technique involves flushing rinsing solutions over equipment surfaces to remove residues, which are then measured in the rinse solvent. In addition to swabbing and rinse sampling, a visual check for cleanliness is an important part of the cleaning validation acceptance criteria. However, it needs to be stressed that relying on visual inspection alone for determining cleaning effectiveness is not an acceptable practice. Also, visual inspection should be performed under appropriate lighting conditions that would enable acceptable inspection of the clean surfaces, whereas boroscopes, fiber optic probes, and other scopes may be employed to inspect locations which would be otherwise hard to inspect. The accuracy of sampling methods, either swap or rinse, relies on the determination of recovery factors by means of recovery studies, which allows for establishing the relationship between the result of a single sample to the total amount of residue on equipment on the sampled area. This is achieved by spiking known amounts of product or API on coupons, which are then allowed to dry before they are sampled to recover the residue. It is important to emphasize that as part of the recovery studies, all involved materials of equipment construction should be taken into account. Similar to swabbing and rinse sampling, visual inspection should be performed on all product contact surfaces, while the level below which a residue is not visible has to be determined through spike coupon studies as well. The influence of time and the impact it might have on the cleaning effectiveness should also be assessed as part of the cleaning validation exercises, both in terms of the maximum length of a campaign in time and number of cycles, and the dirty hole time that corresponds to the time between manufacturing and cleaning. Also, the ingress of microbiological load may be evaluated as part of the clean hole time evaluation that corresponds to the time interval between cleaning and use. Until the cleaning validation campaign has been concluded and the cleaning process deemed validated, or in the event of a cleaning failure, assurance that the equipment is clean and available for further use should be coming from cleaning verifications, a practice which refers to the gathering of evidence through chemical analysis after each batch or campaign to show that the residues of the previous product or cleaning agents have been reduced below the scientifically set maximum allowable carryover level. While validating the cleaning cycle for every product manufactured on site could be an option, this approach nonetheless is of limited practicality at multi-product facilities. This is the reason the bracketing approach, also known as matrixing or family approach, is preferred to streamline the overall validation effort. The term bracketing refers to the science and risk-based validation approach, where different products or pieces of equipment may be grouped together, and validation is performed only on worst-case equipment product combinations, assuming that easier to clean equipment product combinations are adequately represented. Groups of worst-case situations are established with one piece of equipment representing a group of similar or easier to clean equipment, just as residues are grouped by cleaning with one residue representing a group of similar or easier to clean residues. Whenever a new residue or equipment is added to the facility, a new evaluation needs to be performed to determine if new entries constitute a new worst case, which will require a new validation or not. Finally, as this presentation draws to an end, a number of recent deficiencies have been included here for reference. The first example talks to swap locations missing from the cleaning validation protocols and reports. As previously described, worst case difficult to clean swap locations on the equipment range should be determined during risk assessment. The second deficiency refers to the lack of periodic revalidation through cleaning verifications. Successfully validated cleaning methods should be subjected to periodic revalidation in order to support the validated status of the method over the lifetime of the product, process, or equipment. In the third deficiency, that refers to the absence of recovery studies as part of the visual inspection that would determine the level below which a residue is not visible. Moving on to the next slide. The first example refers to the regulatory expectation that companies should consider scientifically justified methods based on toxicological evaluation to define the cleaning validation limits, while the next deficiency highlights the fact that not all types of product contact surfaces were taken into account in the swap recovery studies. The last deficiency example on this slide addresses the fact that taking into account the swapped area, or in this case, the volume of rinse solution in which residue will be measured, is essential in the macro calculation. as that would enable us to relate the result for a single sample to the total residue amount on the manufacturing equipment. On a similar note, the next slide, recovery factors established relate to the result of a single sample to total residue of the sampled area and should therefore have been considered in the macro calculations as well. Moving on to the next one, it is imperative that validated analytical methods can read lower in terms of LOD and LOQ than the residue limit acceptance criteria in order to be able to demonstrate adequate assurance that the residue traces have been effectively been removed from the equipment surfaces. Finally, in the last example, as the last example alludes to the fact that the entire shared equipment train must be taken into account in calculating the corresponding macro levels, and not by doing so, this results in an overestimation of the maximum quantity of product or residue that could end up in a subsequent batch of product, which could potentially have serious implications for the health of the patient. This brings us to the end of this presentation. I have included in this slide the relevant guidelines to the topic of clinical validation, as well as the ISP guide on clinical validation that includes a number of useful examples and information around this topic. Thank you.